So it's my pleasure to introduce my colleague, John Crary. I work with him now at Mount Sinai in New York. And he's going to tell us about AI in diagnostic and research neuropathology, something that's been on people's lips through the whole meeting. He's professor of pathology, neuroscience, and artificial intelligence and human health. That's an interesting juxtaposition. At the Icahn School of Medicine at Mount Sinai, where he was the founding director of the Neuropathology Brain Bank and Research Corps. Dr. Crary has made substantial contributions to approaches to diagnostic categorization of several neurodegenerative diseases, leading the effort to delineate primary age-related tauopathy and others. He is a pioneer in applying deep learning to addressing neurodegenerative disease. His laboratory was the first to apply convolutional neural networks to Alzheimer's neuropathology. In addition, the Crary Laboratory has developed an AI capable of detecting synucleinopathy in peripheral biopsies, which has the potential to rapidly accelerate the pace of clinical trials. His laboratory also developed a powerful algorithm termed histoage, I love this, which is a novel approach to studying brain age acceleration and neurodegeneration. Further, the laboratory has demonstrated that neural networks can extract essentially limitless numbers of features related to the brain in an unbiased way and train on them, learning the most relevant cellular abnormalities for a given symptom or disease. Currently, the laboratory is training AI models to perform complex neuroanatomical segmentation on the cellular level and others that measure brain age acceleration, neurodegeneration, and functional impairment. So thank you for coming. I'm so excited to hear this. We've talked in the hallway a lot about this stuff.
Dr. Folker, thank you so much for that kind introduction, and to the Society for the invitation and the opportunity to present today on AI. Okay, nothing to disclose. These are my learning objectives. So I guess the place to start is that AI is permeating every aspect of our lives. There's no part of society that is immune. It feels like the momentum is so strong, and there's no part of our world that's not going to be affected. Medicine specifically will be impacted maybe more than other fields. Among medical specialties, the data-rich specialties like pathology will be even more transformed. There's a lot of optimism, right, for personalized medicine, for innovation in research, new therapies that are going to come from these extremely powerful approaches. But along with the hype, there's also skepticism and fear that maybe we could be putting ourselves out of a job, or that we may harm our patients. I'd go as far as to say that, or as to quote some leading figures in the field, such as Elon Musk, who has said that AI is probably more dangerous than the atom bomb. Other AI scientists, such as Jeff Hinton, have really raised the alarm bell. So these are things that we have to think about. Here, this chart just shows the explosion of articles in PubMed that have used the terms artificial intelligence or AI. So there's really no getting out of it. Really what I'd like to challenge the audience or challenge the society is to think about what we envision the future of neuropathology to look like. So this didn't happen all of a sudden, although it feels like it did happen like overnight. Really, AI scientists started in the 1950s with Alan Turing and others, and maybe more contemporary scientists. I mentioned Jeff Hinton, the godfather of artificial intelligence, Jan LeCun, Andrew Ng, really putting a lot of effort into developing these technologies. And here we are in the middle trying to figure out how we're going to apply these extremely powerful technologies to our workflow. Some may envision this post-apocalyptic kind of AI apocalypse, where, maybe not the Terminator, but maybe this artificial generalized intelligence will get rid of like the human race. But I really think it is in our power to envision maybe a more utopian future where we're using AI smartly, wisely, with all the ethical considerations taken into place. This is probably the most important slide that I have to show. Kurt Langlitz at Stanford in response to a tweet. So this is a modification. Will AI replace neuropathologists? No, almost certainly not. But neuropathologists who use AI will replace neuropathologists that do not. I really believe this, and so I've just gone 100% into trying to understand and learn AI, apply it in the lab. We really haven't figured out, it really hasn't emerged in the clinic yet, but there's lots of efforts to deploy AI algorithms in practice. And so we'll talk about that a little bit. So let's dig in. First thing that you need to know is a little bit about the kinds of models, AI models, and some of the terminology. So artificial intelligence, that really refers to software that mimics human intelligence. And there are different kinds. There's expert systems. Those are basically human-curated data sets or algorithms that rely on a human. Machine learning is different. That's when you create software or an algorithm that learns on its own from the data that you provide. I think it's really important to understand that the AIs are not that smart yet. The data that we give them has to be highly structured, highly organized, and there's lots of problems with the data. We'll talk about more in terms of bias, but this is actually critical. There's lots of AI algorithms that have been around for really decades. I'm going to talk a little bit more about digitization of histology, but other disciplines, other parts of medicine, genomics, molecular, have been using AI algorithms for decades. Radiologists who've had all of their MRIs and CT scans digitized, they've been able to apply AI algorithms for a long time. Just simple regression methods are considered machine learning. I'm going to talk a little bit about convolutional neural networks because they're probably the most important AI algorithm that we need to know about. But there's a concept of deep learning, which is also kind of new. Because of the expanse in our ability to store data, the hard drive space, and the new computer processing units, the graphical processing units, we can crunch large amounts of data that we could never do before. So the AI networks are getting bigger and bigger and bigger. We call that deep learning, just because the AI is ridiculously large, with billions of data points within the network. So deep learning is really the thing that's led to the exciting innovations in the past couple of years that we've talked about. So this is also kind of important. How exactly are we going to apply machine learning and AI to neuropathology? There are basically two types of tasks that we would use it for. One is a classification task, or simply like a diagnosis. Based on the data that gets fed in, for example, a whole slide image, you may try to detect something. So, a fungus or, a virus or amyloid plaques or tangles if you're working in my lab. There's also these kind of prediction algorithms. You can think of more of like prognostication. You might hear AI scientists talk about inference. They love to use that term, and it's kind of ambiguous, but inference is basically prediction, could be going backwards or forwards, right? So if we work with autopsy tissue to make a prediction about whether that patient had dementia, kind of doesn't make sense because they're already dead. So we would make an inference about something that might have happened in the past. So inference basically means prediction, but can go both ways. And regression is kind of a synonym. So some kind of prediction task might be, the survival of a patient based on their glioma, their histology, or maybe integrating histology and molecular features. So when you start to take a step back and just start to think about like the neuropath workflow, there's so many points where AI could be useful. Even before the specimen arrives in the pathology laboratories, our partners, or the clinicians, are going to be working. They're going to be pulling data from the clinical record just to try to figure out whether a biopsy is indicated before we even start. Once we get the specimens, they're going to be the slides are going to be digitized. So you could imagine, for example, triaging cases. You could have them pre-screened by an algorithm that maybe figures out, what you should prioritize. So if there's a vasculitis that was unexpected, you could maybe do that first in the morning before the others. So case triage is important. Disease classification, so these image recognition tasks, I'm kind of jumping around, will lead to advances in personalized medicine. Reporting, using language models. We hate structured reports, right? So if we could use the language models, that might be able to help us in more efficient reporting. Quality control. Extremely important, valuable. Integration with other medical data. So not just, looking at an H&E, but you could imagine layering in clinical, molecular, other types of biomarker data in terms of diagnosis. And AIs are extremely good at detecting patterns in large data sets. So maybe something that would not be simple for a neuropathologist on their own. The research possibilities are really limitless. So, and of course, training and education, which I'm not going to talk too much about. So this is really the thing that is probably the most exciting to me. We have all of our images, all of our specimens on glass slides, and they're sitting in drawers and warehouses all over the place. And we know that these H&E slides are data-rich, and also our immunostains. There's tons of data that's just sitting in these slides that we don't have access to. So with these digital whole slide images, we're going to unlock all that data, and it's going to lead to amazing things. For the first time, we're going to be able to apply machine learning algorithms to those specimens, and we're expecting great things to happen. So I'm going to try to explain a little bit about how these algorithms work. So really what's happening is the AI scientists are trying to build a virtual brain within a computer. So they'll create some kind of construct that represents neurons and synapses, and it's essentially, at the heart of it, just a spreadsheet, right? A very large matrix with pointers to other nodes within that spreadsheet. And you can think of each one of the cells in the spreadsheet as a neuron, each one of those pointers as a synapse. And then they start showing pictures or other types of data to this network, and they adjust the weights of all the synapses. And eventually, when you adjust it just right, you can get the network to perform in really interesting ways. So I love these. You can go to the internet. You can find all kinds of funny classification tasks like this. This was very, very difficult for a computer to do, to differentiate fried chicken from your labradoodle when I started. But now we can just crush these sorts of tasks. It's really quite amazing. So the convolutional neural networks, so what I mentioned before, the most important for computer vision algorithms. And let me just say, of course, we're talking about artificial intelligence. There's language models and models that are based on DNA. For me, this is where the biggest advancement is going to come, because we're going to be able to put numbers on images, quantify, basically measure morphology, which we've really never been able to do very well before. So we're going to move forward very, very, very fast in this space. And so this type of network is especially designed for image data. So it's inspired by the cytoarchitecture of the visual cortex. So it has layers. The first layer of this network contains within it hard-coded structures that detect edges, shapes, and textures, types of things that the visual system does as it processes information. Then there are additional layers that are kind of hidden layers within the middle that kind of play around with those structures and link them together into kind of higher order concepts. And then there's an output layer at the end, which kind of tells you, is this a dog, a cat, or whatever you've trained on. So these CNNs, they learn from the data that you give them. They're very accurate. They excel with image data because of the way they're built. They can pretty much classify anything, dogs, cats, children, crossing the street, crumpled up pieces of newspaper or whatever. And so these are extremely useful for, say, self-driving cars, face recognition, so on. You could imagine using them for detecting different types of anomalies or lesions within tissue, or mitotic figures if you're working in cancer, or plaques or tangles, so on. So one of the things that's extremely exciting for us is that you can use them for large-scale studies, potentially. say you want to count your tangles and do a correlative study in 10,000 cases, the entire NAC data set. Like, we could potentially do that with this sort of algorithm, which there's no resident that I would ever ask to count all the tangles in 10,000 cases in all 12 brain regions, whatever. Okay, so here's a fabulous example of using a convolutional neural network in neurodegenerative disease. And I just want to acknowledge that most of the examples that I'm going to be using are related to neurodegenerative disease because that's what we work on, but there have been similar sorts of studies in glioma and other contexts. So this study, which has been discussed previously at this meeting from the Duggar group, developed a CNN, and this was a neural network that was trained on over 70,000 images of amyloid plaques that came from 43 different whole slide images. This, I think, really just showed us how we could apply in a brain bank setting a pipeline to pull out these features in a reproducible, robust, and much more rigorous way than the kind of semi-quantitative scores that we usually use. This is another example of a way you can use a convolutional neural network in a kind of different, more creative way. So this is a group at Stanford with Tom Montine, he et al. In this example, they took Luxol fast blue counterstained H&Es and did a pseudo immunohistochemical stain. So they were able to use neural networks to predict, based on a large training data set of paired H&Es and immunos, whether there was going to be pathology there. So you could imagine how this might be useful in maybe settings where you don't have the budget to pay for immunostains, or if you want to do different sorts of analysis, cross-comparing different pathology. So there's a lot of information in that H&E that we can potentially pull out. So now here's an example of a kind of an inference task. This was done in my laboratory. Andy McKenzie, who's here at the meeting, was a research track psych resident. And his question was, well, can we rather predict whether somebody has cognitive impairment based on an H&E stain? So we know that it's really hard for pathologists to do this, right? Plaques and tangles don't correlate perfectly with dementia. So using a multiple instance learning pipeline, he was able to train an AI that could do just that, could predict cognitive impairment. Now, it wasn't perfect. It's a small data set. He got up to about 0.65, which is better than chance, but it was also better than what I could do, which is kind of amazing. But what was really amazing about this pipeline is that you can use the signal that the AI is using to make that inference to map the cells and the cytoarchitectural changes in the tissue that were responsible for it. So let me tell you what I mean. The way that this algorithm works is it takes the slide and it breaks it up into patches. And then in each one of those patches, it runs it through a big convolutional neural network. Here we use something called ResNet, and mapped thousands of features onto each patch. So after that first step, then the AI goes through it and it tries to extract the features on each of those patches that are most strongly correlated with the label. So here we were using cognitive impairment as the slide level label. We didn't have to go in and circle all the plaques and tangles. We just said, here's a slide from somebody who has cognitive impairment. So let me just check the time here. So this is kind of like, if you think of training an AI to find a needle in a haystack, this isn't showing it lots of needles. This is saying, here are two haystacks. What's different between the two of them? And so what it does is it can find the things that stand out. You might say, okay, here, look, this is a needle that was in that haystack that you didn't, that you were looking for. So yes, that's what's different about this one. But it'll find other things. There was a mouse in the haystack. There was a boot in this haystack. It can find things that are maybe more subtle that you might not have noticed right away. I like to say, well, it could pick out the species of hay and tell you, oh, this is a different type of hay. And you can say, oh, wow, I understand that. And it can also pull out features that are beyond human comprehension. It can say, here's an embedding, which is a type of feature that's so multidimensional that it's beyond your capability of understanding, but it's different between your two haystacks. And maybe you could use that to discover genes or whatever if it's correlated with the clinical symptom that you're interested in. So this is really this idea of histomics. the ability to not just look at one feature at a time, but to look at everything that's in the slide all at the same time. So this is an extremely powerful algorithm, and to me, this is kind of the killer app, I think, for AI and histopathology. So this is worth learning. We were able to train, using a similar approach, an AI that could predict age in a hippocampus. So if I gave you an H&E from somebody's brain and I said, how old is this patient? You probably wouldn't be able to get very close. I don't think there's a system for doing that. Gabe Marks, who was a master's student in the lab, said, well, why don't we try doing this? The reason why it's so valuable is because you can come up with a metric for age acceleration. So aging is a confusing but important research topic. There's chronological age, but there's also your biological age. It seems like some people seem to age faster or slower than others. And why is that? Well, genetic factors and environmental factors all kind of conspire to either protect you or accelerate your aging. Different organs, different populations, different species age at different rates. Why is that? And we really want to be able to study it. Classically, we use methylation to predict people's age. But Gabe said, hey, can we do a histo age? Can we just use that H&E slide, pull out all of those features? So you don't just look at tangles or plaques, you look at everything and holistically. And the AI in the first pass was actually pretty good. It got within 5 years on average. And we thought this was going to be extremely useful. So we teamed up with, we have been teaming up with Dr. Ann McKee at BU. And we asked, well, if you have repetitive head injury, if you've been exposed to contact sports, you've had repetitive concussions, what does that do to your histo age? And we found in this group of elite athletes, NFL players and others who had played American football, that the AI predicted they were 10 years older on average based on their brain features. So this is a completely unbiased approach that we think is going to be very useful for dissecting out environmental and genetic features that can accelerate or protect from aging. And then using that, using those features, I was going to describe this before, you can map the risk back onto the slide and create these heat maps. So that's called attention analysis. The top row there shows the acceleration, and all the way on the right, the attention map is really focusing, you can tell, on the white matter. One of the things that is really important, I'm going to talk about it more in the next slide, is what network you use to extract the features. So ResNet was trained on the world. This is Fei Li at Stanford's ImageNet Consortium. They got pictures of, lots of pictures of pets, cats and dogs. It just so happened to be a lot posted on the internet, but regular, real-world things. It was not trained in the microscopic world. It still kind of works a little bit because it guesses. It thinks that the cerebellum is a leaf. It thinks that neurons are trees. It thinks that tangles are insects. So you can use that, but it hasn't been trained on the brain. And it hasn't been trained in the microscopic world. We tried using a CNN that was created by Gabriele Campanella, who works in Thomas Fuchs's lab at Mount Sinai. It was trained on 450,000 slides from our service. So we have about 10 petabytes of digital whole slide images that are now sitting on the supercomputer at Mount Sinai that we can use for training algorithms. And you can see how it really tightens up that association. The histo age metric is getting better, and the separation between the CTE cases and the controls is getting better. But one thing that is disconcerting, or maybe interesting, depending on your perspective, is that the attention analysis shifts from the white matter to the blood vessels or to the leptomeninges. We think it might be because this foundation model that Campanella trained was trained on every organ. It wasn't focused on the brain. There's lots of blood vessels in every other tissue. So maybe it got really good at figuring out what's going on in blood vessels. So anyway, what is this foundation model? What is this concept? So the idea of a foundation model is you train on lots and lots of random types of data, maybe not your thing specifically. So for example, ChatGPT is just trained on language in general, lots of data from the internet. DALL-E trained on lots of images. And then you can tune it to your task, and that's called transfer learning. So these foundation models have become really popular. We think there's lots of opportunities in language and vision. There are some technical considerations. The architectures are changing. Broad applications in healthcare, law, education, as you can imagine, for these types of models like ChatGPT. There are risks. I think it's really important that we understand that the models are biased. So inequity, misuse, they tend to hallucinate, they make stuff up. So these are all problems that we have to deal with. The ethics, and we need to think really carefully and long about those types of biases. One thing that we're concerned about is this idea of an emergent capability. So we don't actually know what these models are capable of. Will they acquire abilities that we didn't know they were capable of? We don't really understand how they fail, and so on. So here's an example also from Stanford, the Hong et al. With Tom Montine. This is a visual language model. So this model is amazing because it can take text and generate histological images, or it can take images and convert that to text. So it can describe what's going on in the tissue on the microscopic level, which is amazing. This was trained on a data set that was pulled from Twitter. I think in the future, for us as a society, we're going to want to think about having large data sets that are curated by actual, neuropathologists who are trying to generate gold standard data. This was a really resourceful way of quickly getting data. There's a bunch of different foundation models that are emerging. This one, GigaPath, I wanted to point out that Microsoft created. The number of images is just huge. Did I put it there? How many? It's a couple 100,000 slides, billions of pathology, 1.3 billion pathology image tiles. So those all came from slides, but it doesn't say how many slides. What was really frustrating about this foundation model is almost no neuropathology in it. They're getting most of their images from clinical services, and we're very much underrepresented. So as a forward-thinking thing for the society, we need to, if we want AI and reliable, robust AI on our service, we're going to have to build these training data sets, or else it's not really going to happen. Most of the models are opportunistic. So AlphaFold, which is probably one of the biggest successes in AI, was built off of the PDB data set, the SwissProt data. It was already on the internet, and so they could just download it. But those are all globular proteins. You're out of luck if you want a membrane protein. So they're starting to think about, well, maybe we have to do some targeted structures so that our models can get better. Just like that, we're going to have to start thinking about building the data sets that we need across all of our subspecialties so that we have these sorts of models that can pick things up. If I just train on neurodegenerative data, if there's something interesting happening that's inflammatory, for example, we may miss it. So here, this image is from ChatGPT. I asked it to provide an H&E image of a neuron in the hippocampus on an FFPE tissue section. So it kind of did okay. It looks a little bit like a neuron, maybe more like an astrocyte if you ask me, but it seems to be embedded in cartilage or whatever. So I think these things will get a lot better if we can create these data sets. That's most of the work, by the way. It's 99% of the effort and the cost is going to be creating the annotated data sets so we can train the AI. Running the AI models is easy and it's fun. So that's not going to be the barrier. We used to have that barrier at the beginning. There weren't enough scientists who knew how to run these algorithms. That's been solved. So lots of sources of bias, picking the problem, where's the data, what kind of data sets are we putting in there, how we define the outcomes, do the big algorithms, do they, the big foundation models, do they apply to subpopulations? This is all really important. I'm going to end really quickly. Here are some questions that come up a lot. Can I use ChatGPT to write a manuscript? Well, JNEN has instructions now, so no for manuscript reviews. Peer reviewers are prohibited from uploading your manuscripts. That's because it's considered if you want to review a manuscript, you can't upload it. But if you want to use AI to help you, you can to write your manuscript, but you need to disclose it. So, because of all these problems, so this is JNEN's instructions. Springer is different. They don't want you using any generative AI tools at all because of the potential copyright rules that haven't been sorted out in terms of the training data for those. Is it really allowed? So the NIH prohibits you from using ChatGPT for reviewing grants. Once again, it's considered a breach of confidentiality, but you can use AI to write your own grant, but you really have to be very careful. Potential plagiarism, it makes stuff up, so buyer beware. And then I'll end there. Thank you so much.
That was terrific. Thank you so much. I understand much more than I did before. So we have time for a quick question if anyone has one. Bill?
Yeah, thanks very much. That was a beautiful presentation. What advice do you have for the audience members, since we're seeing these more and more and the results of the modeling more and more in research settings? What advice do you have for us to recognize overfitting? To recognize, in other words, something that's really, really good at the training data set, but not necessarily on real-world applications.
Yeah, that can be really tricky. Probably the most important thing is looking at replication across other data sets. Usually the standard is to take your data, take out a holdout for testing and for validation. Because the testing is, the validation is to validate the model, and then the testing is the thing that you're curious about. That's absolutely essential. I have a feeling there's going to be lots of people who are going to come up to me in the hallway and tell me all sorts of opinions about AI, and I will hang out also here.
Thank you.
Yeah.
Well, that was terrific. Thank you all so much for your attendance. That closes the sessions for today, but the diagnostic slide session is tonight at 8, so I will see you all there.